Hey guys, happy Star Wars Day. Got my Star Wars periodic table shirt on to represent. Hope you had a good weekend. Recording this on Monday. So what we're going to do today is move on in our unit to start to talk more specifically about genetics. And you guys studied genetics in 7th grade, and so some of the sections that we do in the notes today we'll be reviewing. We'll move through them pretty quickly, but then we're going to talk about some more complex topics related to genetics. So when we talk about genetics, we are talking about the study of heredity, the study of how traits are transmitted from parents to offspring. And we know that that happens by the inheritance of chromosomes, which contain DNA, which contain the genes. So you may remember from 7th grade, just to talk about the history of genetics a little bit, Gregor Mendel was an Austrian monk working in mid-1800s, and he was sometimes called the father of genetics, and he was interested in studying pea plants. And so pea plants grow very easily, can be cross-pollinated clearly, and had some distinct characteristics that can be observed easily. And so he used these characteristics of pea plants to crossbreed thousands of pea plants and very carefully collect data about the traits that each plant had. And in analyzing that data, he observed that there are certain mathematical ratios between the offspring that were consistent amongst different traits. And that allowed him to establish the basic rules of Mendelian inheritance, is what we call it, 3 basic laws of genetics. Mendel's work really, though, was sort of, it was published, but not really, it didn't have a huge impact. It was sort of lost for a long period of time. It wasn't rediscovered by scientists until around 1900, when scientists saw his work and started to combine that with information that they had gained about genetics and the genetic material in the cell, that allowed them to sort of go back and revisit what Mendel said. One scientist working in the early 19th century, 18th century, early 20th century was Thomas Morgan. Thomas Morgan was an American geneticist at Columbia University, I believe. He came up with the term gene. He did his work mostly with fruit flies, which you see here at the bottom. This is a fruit fly, Drosophila melanogaster, and they are a commonly used organism in genetic research. They have certain mutations which can be observed pretty easily. They go through generations very quickly, and Morgan studied these fruit flies in his lab and basically confirmed that Mendel's laws did apply to animals. He established the term gene and hypothesized that genes are found on the chromosomes, that certain traits are sex-linked, and so he added significantly to what we know about genetics. And as scientists started to study genetics more and more, one of the questions was about, well, how is this information stored in cells, in organisms? How is it coded? What molecule carries the information and passes it down from generation to generation? There was debate about this. Was it proteins? Was it nucleic acids? And what structure were these materials? And scientists are sort of credited with determining the molecular basis of genetics, the molecules and the structure of those molecules, were James Watson, Francis Crick, with help from Rosalind Franklin. Basically, they define the structure of the DNA molecule. They put together a model that showed that it's in the shape of a double helix, and that the genetic code is stored within the sequence of bases of those of that DNA molecule. And this is an X-ray crystallography image. This is from Rosalind Franklin. She was a woman, a scientist that was working, also studying DNA. And this image is a famous image because this image helped Watson and Crick come to their conclusions that DNA has this double helix shape. However, it wasn't really provided by Rosalind Franklin willingly. It was sort of passed from a colleague of Rosalind Franklin who was working with Watson and Crick, and they saw it. She never really got the credit she deserved for contributing significantly to the establishment of the DNA structure. It's an interesting story to read about how women were treated in science, especially at that time in history. So if you have any interest in that, it's something to look into a little bit. So we've already talked really about DNA and protein synthesis early on in the year when we were in our biochemistry unit. So we're not going to go into really a lot of detail here, but just let's kind of review. So DNA is a double helix. It's a double-stranded structure, okay? And the phosphate and sugars, okay, form sort of the sides of the DNA ladder. And then the connecting pieces in between, the rungs of the ladder, are the nitrogenous bases, the adenosine, thymine, cytosine, and guanine. Those are the four types of nitrogenous bases that form the DNA molecule. They are the bases. The order of those letters is what determines the DNA code. A and T are complementary to each other, and G and C are complementary to each other. They always match up in those ways. That is one of the ways that a DNA molecule can replicate. When the two sides of the DNA molecule separate from each other, okay, free nucleotides start to attach, and they can then form a identical strand of DNA. And that's what you're seeing in this animation here on the right, the splitting of the DNA molecule into its two sides. Free nucleotides then matching up on each side. And as this process finishes, what you're going to see at the end is that we have two identical copies of the DNA molecule. So this DNA molecule has been replicated. So this is a process that takes place in the S phase of the cell cycle, the copying of the DNA to form double-stranded chromosomes. The code that's stored in the DNA is found in the nucleus, in the chromosomes. However, it's sort of locked in there in the nucleus. The DNA molecule cannot leave the nucleus. So instead, when one gene is going to be used to produce a protein, it gets copied, and this process is called protein synthesis, of the copying of that gene and then the creation of the protein. So inside of the nucleus, when a gene is going to be transcribed and translated, the DNA sort of unwinds a bit, and the gene, which is again just a section of this DNA, is copied into another molecule called messenger RNA. That messenger RNA can leave the nucleus through these pores, and that messenger RNA is now carrying the recipe for how to build a protein. It can move out into the cytoplasm, where ribosomes attach. Ribosomes are also made of RNA. They're called rRNA, ribosomal RNA. They attach, and that's you see an enlarged view down here. And so the messenger RNA is carrying the recipe, and then the third type of RNA, transfer RNA, starts bringing in, according to this recipe, the correct amino acids. And they start to become linked together to form a protein, a polypeptide. Okay? There's about 20 different amino acids. There are 20 different amino acids that go into making human proteins, And so the transfer RNA brings in the correct combination of those proteins, and they get linked up in sequence until eventually the protein is complete and is released and then folds up into its final shape. Here's an animation. So we have transcription, the first part of the process, copying that recipe onto messenger RNA. Then transfer RNA molecules bring in the correct amino acids to the ribosome where they're attached, forming the protein. So when Mendel was studying genetics, he was studying pea plants. And as we said, pea plants had several easily observable characteristics. So some of the things that Mendel looked at were the shape of the seeds. They can either be wrinkled or they might be round. The color, yellow versus green, of the seed. The color of the pea pod, yellow versus green. The shape of the pea pod, okay, either smooth or constricted. The color of the flowers, the position of the flowers, the length of the stem of the pea plant. So these were all factors that Mendel looked at, and they were all sort of very clear that the flowers were either purple or white. The seeds were yellow or green, wrinkled or round. All of these traits basically just came in two different versions, so they were a good model for Mendel to look at. And so Mendel studied these plants, and what he did is he took pollen from one plant and used it to pollinate another plant, grew the resulting seeds to see the characteristics that he observed. Okay, and he could do this thousands of times, keeping very careful notes. So Mendel came up with several laws that we call the laws of Mendelian genetics. And the first one is the law of dominance. So we're going to use this term allele. Okay. So an allele is just one version of a trait. So for example, hair color, we could say hair color is a trait. And in our genes, we have alleles that result in brown hair, or blonde hair, or red hair. So different sort of versions of that trait. Blood type. There are several alleles for blood type, several versions of blood type. So in Mendel's research, what he found is that some alleles, some versions of these traits are dominant, while others are recessive. And organisms that have one dominant allele, that's the allele that will show through. It will cover up the recessive allele if it is there. Okay. For example, if we go back here, round seeds is the dominant allele, wrinkled is the recessive allele. In flower color, purple is the dominant allele. In seed color, green is the dominant allele, or yellow is the dominant allele, I'm sorry. So when Mendel was studying these traits, he used symbols. Now the symbols, we use sort of conventions. You could really use whatever symbols you wanted to, but we have typically, when we're talking about Mendelian genetics, we often will use the first letter of the dominant allele as the symbol for that gene. And if we're talking about the dominant allele, we use the capital version of that letter. If we're referring to the recessive allele, we use the lowercase. So, for example, when Mendel took a pure tall plant, remember, tall is the dominant allele, so we're going to use a capital T for the dominant allele. A pure tall plant, and he crossed it, okay, with a pure short plant. So short is the recessive allele, so we'll use lowercase t's. We're completing a Punnett square here, a model that helps us predict what the offspring of two parents might be like. We did this in 7th grade. We combine into each box, so big T, little t, big T, little t. And so that's what Mendel found. When he crossed a pure tall plant with a pure short plant, every single offspring was also tall. Okay? And that led him to his ideas about dominance. That what Mendel said is that, well, tall is a dominant allele. If you have one of these dominant alleles, that's what's going to show through. And since every offspring ended up with at least one dominant allele, that's why all of the offspring were tall. So we need to define a couple words here. When we talk about the phenotype of an organism, what we're talking about is sort of how those genes are manifest, how they're displayed, how they come through. Often it'll be physical characteristics. So when I talk about this is a tall pea plant, I'm talking about its phenotype. Its genotype are the genetic makeup, the alleles that it has. So if I were to say big T, little t, that is its genotype. If I say heterozygous, which means there's two different alleles, that's its genotype. If I say homozygous dominant, that's its genotype. It means two of the same dominant alleles. So when Mendel was doing these crosses, he used some symbols. He called the original starting parent generation the P1 generation. Okay. So this would be the parent generation. This is the P1 generation. So when he crossed the pure tall and the pure short plants, the homozygous dominant plant with the homozygous recessive plant, okay, what he found is that the phenotypes were all tall. Then Mendel looked at the results of taking two of these offspring. So these would be the F1 generation, the first offspring. Okay, they're 100% heterozygous, 100% tall. What he did is he would take two of those plants and cross fertilize them. So he took two of these heterozygous plants, big T, little t, crossed with big T, little t. And what he found is that when he completed these test crosses, what he found is a consistent ratio. 75% of the offspring were tall, but there were 25% that were short in their phenotype. Okay, so the short alleles that were present in this parent, they were not shown in the offspring, but they were shown 25% of the time in the next generation. And so this is a pattern that Mendel saw over and over again with traits other than tall and short pea plant, with purple and white flowers, with the green and yellow peas, etc. So this F2 generation, this second generation, always showed this ratio. 25% were homozygous dominant, 25% were homozygous recessive, and 50% were heterozygous. In terms of phenotype, 75% of the offspring in this F2 generation were tall, and 25% were short. So this is Mendel's law of dominance. We know today that many traits do not follow a simple pattern of dominant versus recessive. But in Mendelian genetics, when we're talking about these sort of simplified models, okay, then these laws hold true. There are two other laws that Mendel derived from his research. One is the law of segregation and recombination. This law says that when the chromosomes are separating in meiosis, okay, the two different alleles separate from each other and end up in different gametes. So we just covered meiosis. So we know in meiosis, homologous chromosomes align themselves, okay, during the first division with their homologous partners. Now those homologous chromosomes, each one, this is an example here. So I have two homologous chromosomes. Okay, they're not identical, but during the first division of meiosis, they separate. Okay, that's the law of segregation, that this darker green marker will end up in one gamete, and this green marker with a different allele will end up in a different gamete. Okay, so they've separated, they've segregated. And then they will recombine when fertilization takes place. So the two alleles for a certain trait get separated during meiosis, and then in fertilization they get recombined between two different parents. But each gamete is only carrying one allele for the gene. So when Mendel was looking at his pea plants, we'll come back, there was the genes for tallness, big T and little t, two alleles for height of the plant. During meiosis, they got separated, those two alleles. And in fertilization, they recombined. And then the third law is Mendel's law of independent assortment. What this law says is that the alleles for different traits were separating independently from each other. So what Mendel found was that the gene for tallness and the gene for seed color and the gene for flower color all were separate from each other. They weren't inherited together. They were independent. And that was because those traits were found on different chromosomes. The genes for those traits were on separate chromosomes. And so when two different genes are on different chromosomes, they'll be inherited independently of each other. They won't be linked together. Now, if they were on the same chromosome, then they might be linked together because chromosomes are the unit of inheritance. So this diagram on the left side shows us this idea of the law of segregation and recombination. Okay, the big T and the little t alleles during meiosis get separated from each other. Same thing here. They're segregated into these gametes. So some of the gametes have the dominant allele, some of the recessive allele. Then they recombine when those gametes come together in fertilization. That's segregation and then recombination. Okay, and here we see the concept of independent assortment. A gene that's found on this chromosome will be inherited separately from a gene that's on this chromosome, because when these chromosomes line up in meiosis, okay, the pattern they line up, like this is a very simple model, there's only 4 chromosomes total, but they could align in this way, or they might align in this way. So we have the red chromosome on the left, blue on the right, red on the left, blue on the right. And if that's the way they line up, when they produce gametes, this gamete's going to get both of those red chromosomes, this gamete's going to get both of those blue chromosomes. However, if they align this way during meiosis, because it's just random how they align, this gamete is going to get a red chromosome and a blue chromosome, and this one will get the blue and then the red. So this is just 4 chromosomes. Imagine 46 chromosomes. There are so many combinations of how those chromosomes could line up that basically you have each gamete unique from all of the other gametes. So those are Mendel's laws. Another concept that Mendel used was this idea of a test cross. So if you have an organism that shows the dominant allele, like this purple flowered pea plant, that's the dominant allele. But if it's the dominant allele, I can't be sure of what its genotype is. The genotype could be big P, because that would result in a purple flower. Or it could be big P, little p, which would also result in a purple flower. So the question is, well, how do we know what it is? Is there a way to tell? The way that Mendel told is by doing what's called a test cross. Test cross can be used to determine the genotype of an organism showing the dominant phenotype. What you do in a test cross is you cross this dominant phenotype plant with a recessive plant or organism. And if you cross this dominant phenotype with a recessive phenotype, if this plant is homozygous dominant, big P, all of the offspring produced should also be purple. But if you cross this dominant phenotype with the homozygous phenotype and 50% show the recessive trait, as in this case, then you know it's heterozygous, this pea plant, by doing that test cross. So you do the test cross, you look at the offspring. If they're all showing dominant phenotype, we can infer that the parent is homozygous dominant. If you do the test cross and there's a combination of the dominant and the recessive phenotype, then we infer that this parent was heterozygous. It's the only time you're going to end up with these white flowered plants is if the original parent were heterozygous. So Mendel's work involving pea plants, as I said earlier, was fairly simple in that those traits Mendel was studying, they were clearly either dominant or recessive, with sort of no in between. You know, when a plant had the big T allele, the tall allele, and the short allele, the offspring were tall. Okay, the result was tall phenotype. There wasn't sort of in between. But that's not always the case. We call these other cases of inheritance intermediate inheritance. It's when heterozygous individuals are different from the homozygous individuals. Okay. So in intermediate inheritance, there's sort of two categories that we talk about. One is incomplete dominance. Okay. An example of this is when you have a dominant and a recessive allele, they both contribute. There's sort of a blending of traits. So, for example, a common example for incomplete dominance is snapdragon flowers. So let's look at that. So we sometimes, when the convention is that we use capital letters for both versions of the trait. So here I have snapdragons. This is a pure red snapdragon, big R, big R. This is a pure white snapdragon plant, big W, big W. And so when we cross those two, what we end up with, all of the offspring are heterozygous, but they show a blend between these two phenotypes. If you cross a red snapdragon with a white snapdragon, the resulting flowers are all pink. Okay, there's incomplete dominance. Red is not dominant, white's not dominant. Okay, there's a blending of both of those traits. These Andalusian chickens are another example of that. Okay, we have a black chicken and producing offspring with a white chicken. Okay, these are both homozygous for black, homozygous for white, but when they produce offspring, which are heterozygous, they're actually gray. So they're showing that in-between blending of traits when they're heterozygous. Along the same lines, codominance is when both alleles get expressed. So not so much in a blending, but they're both being expressed maybe in different parts of the organism. The go-to example for this are these roan cattle. So these roan cattle, they can be pure red, big R, big R. They can be pure white, big W, big W. When the individual is heterozygous, some parts of it are displaying the white phenotype, some are displaying the red phenotype. So you have this sort of spotted look to it. Okay, so here's an actual roan cattle that's obviously heterozygous. Some areas the white alleles being expressed, in some areas the red alleles is being expressed. Sometimes we can show it using symbols like this, CW, CW, or you could just use R and W. There's also a situation in which it's not just two alleles. It's not just big T, little t, or big W, big R. But there might be more than two alleles that are in play in a characteristic. Okay. And so multiple alleles, an example of this, probably the most common one that we talk about is blood type. Okay, so there's not just two versions of blood type. You probably know this. You might be type A blood, type B blood, type O blood, AB. And the reason is because in humans, for the ABO blood group, there's actually 3 different alleles that a person can have. Okay, and so there's the A allele and the B allele. Those together are codominant. Okay, they can both be expressed. And then there's another allele, the recessive. We indicate it usually with a lower I. Sorry, I don't know if you could hear that, but they're mowing the lawn outside and my neighbors. So what this looks like, so we write this typically with these letter I and then either an A and a B or a lowercase I for the recessive. So if a person has the IA alleles, they would have type A blood. Or if they have an IA and then the recessive little I allele, they still have type A blood. Type B blood, same situation. If they have 2 IBs, they have type B blood. Or if they have an IB and the lowercase I, then they also have type B blood. If they get an IA and an IB, then because those 2 alleles are codominant, they're both expressed. So that's what it means when a person has type AB blood. They're expressing both of those alleles. And then the last situation, if a person gets 2 lowercase I of those recessive alleles, then they have what we call type O blood. So we could show a Punnett square illustrating this. And we do it in a similar way. It's a little more complicated because we use the I's in here. So this would be an example. This would be one parent that has type O blood, little I, little I. This parent has type AB blood, IA and IB. We combine and look at our offspring. Okay, and again, 50% of the offspring would have type A blood, 50% would be type B blood. Another example of this multiple alleles is with these rabbits. Okay, so the rabbit fur patterns are inherited in a pretty complex way. Okay, there's several alleles that determine the fur color of a rabbit. So there's the big C allele, okay, which is for brown fur. There's the CCH, okay, allele, which is recessive. So the full brown is dominant over all of these other versions. And then sort of in decreasing dominance, you have the CCH, the chinchilla fur, the Himalayan fur color, or the albino fur color, which is the most recessive one. And so depending on which combination of those 4 alleles a rabbit gets results in all these different patterns of fur color. So again, it's just a more complex pattern of inheritance than the simple dominant recessive. We also know that there are certain traits that are sex-linked, okay, or that are linked together. And so sex-linked traits are traits that are carried on the sex chromosomes. So we'll talk about this in a few minutes, but humans have 23 pairs of chromosomes. Okay. And one of those pairs of chromosomes are called the sex chromosomes. A male has an X and a Y sex chromosome. A female has 2 X chromosomes. And if we look here, so sperm cells can either be carrying the X or the Y. So if you have an X-carrying sperm cell that fertilizes an egg, then the result is in a female sex chromosomes, XX. If it's a Y-carrying sperm cell that fertilizes the egg, then you have a male sex determination. The chromosomes are different. The Y chromosome is actually a tiny little sort of piece of chromosome, carries very little genetic information. The X chromosome is much larger and carries more genetic information. So sex-linked traits are often carried on this X chromosome, okay, and they're often recessive, but the Y chromosome does not carry those sex-linked chromosomes. And so typically what we see in this pattern is that recessive sex-linked characteristics are more common in males versus females. And that's because, again, males have only one X chromosome. So for them to inherit a recessive disease or a recessive trait, they only need to inherit one copy. Females, because they have two X chromosomes, in order for them to exhibit this recessive trait, they have to inherit two of those recessive chromosomes. So it's easier for a man, or it's more common, I should say, for males to inherit these sex-linked characteristics. Examples of things like this, hemophilia and colorblindness, these are sex-linked traits. So let's look at an example here. If we're trying to show a Punnett square with a sex-linked trait, what we'll do is we'll include the sex chromosomes. Here we have X and Y. So here's the male, X and Y. Here's the female, XX. And then above those, in sort of a superscript, we will write the allele that chromosome is carrying. So here we're looking at color blindness. So here we have a father with normal vision. So his sex chromosomes are XY, and on his X chromosome he's carrying the normal allele. The Y chromosome doesn't carry an allele for sex or for color blindness. So we take that father, we put their chromosomes here, X, big C, and Y. Then let's look at the mother. So the mother, she's a carrier for color blindness. So it's the mother, so she has two X chromosomes. One of those X chromosomes is carrying the normal vision allele, and then the other is carrying, this small c represents the colorblind allele. Okay, so she would not be colorblind because she has a normal vision allele here, but she is a carrier. So we put her genes on the side of this Punnett square. Big X, big C, little x, little c. We then complete our Punnett square as we normally would. And here are the results. We get X big C, X big C. So this would be a girl that has normal vision. Here we get X big C, Y. That would be a son with normal vision. Down here we get X big C, X little c. So again, that would be a daughter, and she'd be a carrier. And then finally we get X little c, and then we get a Y. This is a son that is colorblind. Okay. So that's how we show a Punnett square with a sex-linked characteristic. We also know that if traits are, the alleles for traits are close on the same chromosome, then they may be linked together. They may be inherited together. And the reason for this is because of crossing over. So, when crossing over happens and chromosomes exchange pieces, it happens at sort of a random position on the chromosome. So if you have the alleles that are very close to each other on a chromosome, for example, let's look at this example. Here we have a chromosome. We have two genes, gene 1 and gene 2, and they are very close on this chromosome, we would say that they're typically linked, because in order for them to be separated from each other, crossing over would have to happen right in this little area splitting those two genes from each other. If we look at this example, if the two genes are far apart on the chromosome, they're not linked. It means crossing over could happen in many different places between those two genes, and they could get separated from each other. And finally, if they are on completely opposite chromosomes, obviously they're not linked. And so scientists use this concept to sort of map out where different genes are located on these chromosomes. Okay? So they did that by looking at how often do these traits recombine with each other. And that can be used to map where on the chromosome they are located, where they're stored. Another thing that you study in 7th grade, we're going to come back to again this year, are pedigree diagrams. So pedigrees show basically a family tree, and it shows the relationship between people as well as which individuals have certain traits. Okay, so we're usually focusing on a specific trait in a family when we look at these pedigree diagrams. So the symbols, a square represents a male, circles represent females. Connecting directly side to side means they had children. Children go beneath them. Okay, children get connected up and over to each other, siblings. We can show twins. This is identical twins. Okay, this is non-identical twins, fraternal twins. Okay. We can also shade in. So shading in the symbol typically represents they have a certain trait. Not shading in means typically they don't have a certain trait. And half shading it usually means that they're heterozygous. So when we trace a family through a pedigree, it can help us to distinguish sort of the inheritance pattern of a trait. For example, when one parent has the trait, the other parent does not have the trait, okay, and all of the offspring, okay, are showing the dominant phenotype, then that can tell us about the inheritance of that pattern. We see sex-linked traits might be more common with males. So there's ways of exploring the pattern of inheritance by looking at these pedigree diagrams. Okay, and so we'll finish up here by talking just about mutations. So we studied this back when we talked more about molecular genetics, but a mutation is a change in the genetic material, okay, an unplanned change that when cells are replicating, there can sometimes be mistakes in the copying of the DNA. And so mutations that happen in body cells, okay, only affect that individual. So the only time a mutation can actually be passed on to the next generation is if it occurs in the gametes, in the sex cells. That's called a germ line mutation. So if there is a mutation to happen in a sperm or an egg cell, then that would be present in the embryos and all of the cells of that embryo. A mutation in a normal body cell, what we call somatic cell, only affects that individual. So if a person had a mutation in skin cells because of ultraviolet exposure, that mutation will only affect those specific cells. It's not going to be passed on to the next generation. There are also mutations in chromosome number or structure. And typically this results in very pronounced effects because a single chromosome contains genes for many, many traits. So if there is an extra chromosome or a missing chromosome, that will usually have significant effects on the individual. Okay, and there's some examples of this in humans that you're probably familiar with. Okay, one type of chromosome alteration that happens is called non-disjunction. So typically in meiosis, when the chromosomes align with their homologous partner, they separate and end up in different gametes. But sometimes, rather than separating, two of those chromosomes can go to a single gamete. And so that gamete, that sperm or egg cell, ends up with one extra chromosome. And if that sperm or egg cell is used in fertilization, that can lead to an embryo that has an extra chromosome or a missing chromosome. By far the most common chromosome alteration that happens in humans is having an extra 21st chromosome. So when a person has an extra 21st chromosome, it's what is classified as Down syndrome. Okay? And having that extra chromosome results in several differences that are in physical and developmental areas that affect an individual. And so all of those sort of manifestations of that extra chromosome is what we call Down syndrome. It increases with age, maternal age. So as a woman gets older, the likelihood of having a child with Down syndrome increases. But there are other types of non-disjunction abnormalities as well. Klinefelter syndrome happens when a person is born with an 2X and a Y chromosome, or Turner syndrome when a person's only born with a single X chromosome, and these lead to different effects depending on which chromosomes are. Having an entire set of chromosomes, okay, typically doesn't ever happen in humans. If, for example, a sperm cell had a whole extra set of chromosomes, it wouldn't successfully fertilize an egg cell. Okay, and so there would never be an offspring developed. But it does happen in plants. Okay, plants could have an extra set of chromosomes or even two extra sets. Also, sometimes pieces of chromosomes can be broken off from one chromosome and attached to another non-homologous chromosome. That's called a translocation. Okay, and that's shown here in this diagram. We have a translocation of a part of one chromosome being attached to another chromosome. So chromosome 4 and 20 are swapping pieces, resulting in an abnormality. Here we see an example. This is a karyotype, a picture of the chromosomes of a person with Down syndrome. Okay. And again, what we see is 3 chromosomes here. So in all of the other pairs of chromosomes, there's 2 pair. There's 2 chromosomes, or 1 pair. But then we get down to the 21st chromosome, and there's actually 3. And so because, again, there's many genes on that 21st chromosome, it leads to a whole host of difficulties in terms of physical and developmental development. Here we see again an example of having that 2X and a Y chromosome, okay, another type of chromosome abnormality. And so those pictures are called karyotypes. They were traditionally made by doing an amniocentesis or extracting some cells from the amniotic fluid of a pregnant woman, and you could take those cells and then take a picture of the chromosomes and then arrange them by size, and by looking at those chromosomes you could tell if it's male or female. You can tell if there are any chromosome abnormalities and so forth. So here you have some examples. Nowadays it's done using a computer, but here we see, for example, this is a man with Klinefelter syndrome having that extra X chromosome. Okay. So here's a normal karyotype where we have our 22 sets of chromosomes and then the sex chromosome, either XX or XY. Here we see Down syndrome. It's also called trisomy 21. It means 3 21st chromosomes. Okay. And here we have Turner syndrome, where you have just a single X chromosome as a sex chromosome and no Y chromosome. So when mutations happen, a change in the DNA of an organism, okay, it leads to variation in the offspring. Okay, typically mutations are random. This is more common than one of those chromosomal alterations we just talked about. And I think we talked about this back in genetics, but in gene mutation, an extra DNA base can be added in, or deleted, or substituted, one letter for another. And so they can have an effect on the phenotype, or they might not have an effect. It depends on where in the DNA strand these mutations take place. There are certain things that can cause mutations to take place, things like radiation, X-rays, UV rays, certain chemicals and toxins can increase the rate of mutations in cells. One example is albinism. So sometimes a gene mutation can lead to a person not producing melanin, the pigment that's found in our hair and skin and eyes, because of having one of those DNA bases incorrect. And that leads to a person not producing those pigments, okay, having very fair skin, fair eyes, and fair hair. Often these gene mutations might be harmful, but occasionally they might be helpful and provide adaptive value. And we know from our talk of evolution that variation is important. Okay? And so mutations are one of the ways that variation arises in the species. And then the last thing we'll talk about is the environment. So the environment often interacts with the genes, and that the sequence of bases in an organism isn't the only thing that determines the traits of that organism. Often the environment interacts with those genes and can alter the expression of those traits. For example, here we have a Himalayan rabbit, which is an interesting example. So the Himalayan rabbit has light or dark fur, and actually the gene for the darker fur is only expressed at low temperatures. So if you take one of these Himalayan rabbits and you strap an ice pack to its back, okay, what will happen is the fur that will grow underneath that ice pack, that dark colored gene gets turned on under those cold conditions, and that fur will develop into this darker color. Plants, okay, typically express chlorophyll is produced by plant cells. That's why they're green. Okay, but when you have plants that are grown and are not exposed to light, okay, that gene turns off for chlorophyll, and they are growing without that green pigment. If we think about identical twins, we know that identical twins might be identical in their appearance in most ways, but there are differences. There's differences in appearance, there's differences in other factors as well, because it's not just the genes. Identical twins have the exact same genes, but how those genes interact with our environment is also a part of how those organisms develop. So scientists often study identical twins to help them get an idea about what traits are purely based on our DNA and what traits are more about the environment or other factors outside of the actual genes that an organism has. All right, so that's where we're going to stop for today. Our next section will be about human genetics, but that's quite a bit of notes. So thanks for listening, guys, and hope you have a great day. I miss you guys, and we'll talk to you later.